welcome to today's lecture. We will continue about comparative modeling. We started discussing Modeler, a suite of programs that is really easy to use on your machine. One of the best ones out there. Let's talk about a different approach. Swiss model, originally different approach that comes today from the Schwede lab. Thorsten Schwede in the Biocenter in Basel. Overall, in 2011, only 45 publications. One, however, over 2,000 times. The H-index is not that impressive, maybe, but the web presence is. Websites that have over 1,000 access points every day. Another point that is very peculiar about Thorsten, he did his PhD in experimental biology, experimental structural biology, X-ray crystallography. So he's an experimentalist turned informatician or computer scientist. And that is his creed today. Now, Swiss model comes originally from Manuel Peitsch. And Nicolas Bouet has been involved in that. That's essentially the first publication of this. And the idea simply was, again, remember, comparative modeling is about finding a template in the database of known structures in the PDB that is likely to have the same structure as your protein, your target, and you simply copy the coordinates. Okay? Can you do better than that? Is that dumb? It's simple. Okay? So you do the alignment, you find the matching positions, you copy the coordinates for the matching positions, and everything else you leave untouched. How could you hope to improve over that? Remember, we talked about Modeler that tries to, in fact, look at clashes, local clashes, that looks at ways to look up proximities or just let me finish my sentence, contact propensities in order to avoid clashes. What else could you do? You could input these really good matches to Modeler and tell Modeler to keep that. Yes, you could use Modeler. No, that is very true what you say. But it's also true that we're talking 97. So we're talking Modeler, yes, had been done by then, but was just one of the many programs available. And Manuel wanted to have a different approach. And one key element about a different approach was speed. Whatever Modeler does, it's modeling, as the name sort of reveals, as looking up a lot of statistical potentials and the like, and has been doing that from the beginning. It did initially, very from the beginning, some sort of molecular dynamics. Here the idea is blast, look up coordinates, done. This is not time intensive. It's very, very, very fast. And, by the way, then later on board came Thorsten Schwede. And Thorsten initially came on board as sort of the expert in structural biology. Manuel has been in the drug industry for a long time, but he really was not an expert on structures as much, clearly, as Thorsten. And one of the hopes to bring Thorsten in was to, in fact, learn how Swiss model would do. Now, here's the website. And the underlying philosophy of Modeler was you want something fully automated for non-expert users, experimental biologists, and this is a very important part of the main philosophy. So the less you do, the fewer mistakes you make. Okay? So ultimately that means you don't do anything, everything is cool, right? Exactly. You do the blast the sheep, you copy the coordinates, and you're done with them. Is it going to work?
Well, for everything in Swissprot, yes.
Why is there a difference between everything in Swissprot and everything in Uniport? Or I assume this is what you meant. Oh, because Swiss model is called
PDB. Let's take PDB. For everything that we have the structure, everything that is similar enough that we can find it by blast or side blast, yes, it's good.
But then how do you handle, again, let's look at two proteins that have 80% sequence identity. They most likely have a similar structure, mostly, right? Do they have all the residues corresponding to each other? No. Do they have identical loops? No, that's exactly where, in fact, the specificity between mouse and human and the like, why we cannot cross-connect these enzymes of us. That is the origin of all kinds of problems. But that means I have a different loop. How do I handle that different loop? Loop modeling is what... Didn't you program loop modeling? But okay, your program didn't work, or there's another program that didn't work. But in principle, you would assume that loop modeling would fix that, right? I mean, clearly it's made for fixing that type of problem. So this should work less well. So that is true, and that is in fact what everybody, including Manuel Peitsch in the beginning, believed. We will get back later to CASP, but the reality is it did not perform as poorly as the authors had assumed, and it became a serious contender just because of this speed and because it performed in a competitive mode. So this added speed, plus it did not do as badly, got it into the field. But then things changed, and they got way more complicated in that now. Lorenza Bordoli is involved here as the first author of that paper. Is really a decade of effort in the lab of Thorsten Schwede. So we begin with a target, find a template, and now we do the automated alignment. But once we have the alignment, we in fact re-evaluate the alignment, we reassess the template selection, we find a way to handle simultaneously multiple templates, and actually we are on the similar level of complexity as Modeler at the end of the day. Now the tool has become something that is very different. It's more like Modeler. Still, it is on the same scale of accuracy now, performance. Those are very comparable tools. Still, essentially at this point there is a difference in terms of the audience they target. While Modeler is more written for people like you. So people who use programs, have a solid background in biology, sort of are computational biologists. The Swiss model continues to be the first interface for people who really have a first go at modeling, so experimental biologist. I thank Christopher Andrew Wilton for some of the slides, but I'm actually going to fast forward too fast to, in fact, present the slides. So that's the template search. Again, that is also like Modeler today. There is no single alignment anymore. We begin here and we go to PsyBlast, and then we do the HH search, the HH Blitz alignments. Now, in fact, Swiss model has switched to HH Blitz for some of the searches. We do template quality lookup, bound substrate lookup. So we in fact do a text mining to see whether the target that I have is bound to something. If that is bound, that will select the type of X-ray structure, the bound or not the bound form, depending on that text analysis, interpro annotation, secondary structure prediction, and so forth and so on. So there comes additional information, and all of that is being processed, in fact, to align. The loop is modeled, sidechain placements are changed, and then there is an energy minimization at the end of the day. Ultimately, this looks very similar to the slide that I showed you for Modeler. The details are different, the way the information is used, many things are different, but it grew into an alternative package of an alternative level of accuracy. One of the particular aspects of Swiss model, in some sense, is the graphical interface. They spent quite some time on, in fact, getting that to be working for many people. Other comparative modeling methods, and I'm fast forwarding through them. HH-Pret, Zöding, essentially, I'll get back to that, 3D jigsaw, what if and coma. So just the main differences here, HH-Pret, Zöding, Lupas, the main difference in principle to all of the others is the way the alignment is built. Now this is an HH Blitz, HHM, proper profile alignment. It gets many details right, but at the end of the day, it does exactly what you said Swiss model should do. It runs Modeler. So the big difference between this final step and the result that you get out from Modeler is in the way the alignment is done. So that's the major, and there are some, so alignment is done ultimately also including re-ranking, finding different targets. So that's all part of the HH-Blitz and refined HH-Blitz procedure. That's the major breakthrough. And in fact, you know, this creates results that are better than what you get if you ran modeler from the default template. So that is HH-Brit, 3D jigsaw. What would that do? You know what a jigsaw is? Jigsaw puzzle. Comes from Paul Bates and Michael Sternberg. Okay, that's a jigsaw. So, any idea what you do? Yeah?
Fragment assembly?
Exactly. Fragment matching. So now by having a library of fragments, you can refine both the way you redo the alignment, you do the target finding, and you do the assessment of the model. That essentially is a target, is a template, or a fragment assembly in the background of a structure that you already have. It's some world in between the two extremes. So again, let's just one step back here. Fragment assembly. Essentially the idea now is, rather than finding a protein that has a similar structure, I find pieces that have similar structure. The whole thing may not be in the PDB, but every single piece most likely, bless you, is. So every piece of a helix is somehow there, right? Every strand somehow is there. They may be a little bit more twisted, but in principle, the twist is not assessed by what you find, because you find only one pairing of one set of strands. So there is not much of a twist in there. And then you build up. That is one way, in fact, in which people try to predict structures from sequence. The problem really is that even for fragments, the problem still is a complicated one. But now, this is reducing the problem because you fit into a world of known structures. Okay? You no longer try to predict from sequence structure, you just try to predict how that fits to which of the models best. So again, identify similar fragments, assemble the fragments to the structure that you know. What if? That's a method from Gerd Friend. What if the interface comes from Yazara, Elmar Krieger, and arguably, so if you want to make a nice structure, go to Yazara. I mean, you know, this is really very, very, I feel at least, very nice. So here's the background idea. And that comes from Gerd Friend. His original idea, and that also explains the name, was always when you look on a screen at a structure, okay, what if you wouldn't have a positive charge there? Or what if you replaced the lysine by an argine? How would that affect the structure? So imagine I could just go in with my hand and take that out and put something else in there. So what if originally was meant to be a modeling tool? He started programming on that in the 80s. In order to answer the question, how can I do protein design? What effect would a particular change have on that structure? Now, the moment you build up a program that in fact can assess the sort of energy, the spacing, the bonds, the contact potentials, and you in fact also have a tool that does comparative modeling. And that in fact is entire, the what if suite with the interface of Yazara, it is useful for many people. What if it's just one of these monsters that come out from a different time for you guys? So, you know, what if plays chess? Just one of the many things what if does. There are tons of Easter eggs in what if that only Gerd Friend may remember. Most likely he remembers them all. But it's just one of these giant programs. But quite remarkable in terms of its achievement. Coma. Now, Coma comes out from Czesław Węchlowicz in Vilnius, Lithuania. And the major issue that Czesław, in fact, did initially is he improved over multiple alignment. He had, for instance, introduced a position-specific gap penalty and a global score. And initially, the way he optimized all of that was really visually. He started, in fact, playing around with PsyBlast, he was one of the first to use PsyBlast in both directions. We discussed about that 2 weeks ago, two lectures ago. And this was the first time this was, in fact, used to really build models. His alternative alignment programs make Coma become a very, very good contender in terms of predicting structure for comparative modeling. So the latest, in some sense, is going one step further toward fragment assembly, toward structure prediction. ITASER is one of the leading tools here from Yangchang, University of Michigan. And that, in fact, is essentially, you really look at fragments, assemble fragments, predict the conformation of fragments, and map that to 3D structure one step further than 3D jigsaw, because there's much more MD in there. Still pretty easy to use, and is in fact one of the most accurate things. And this slide is Cécile, Nepa, and Pip. For those of you who do not speak French, that translates to "this is not a pipe," obviously. René Magritte, and the slide comes from Thorsten Schwede. Point 1, a model must be wrong in some respects, else it would be the thing itself. The trick is to see where it is right. And that is a statement that I feel is very, very important. So whatever methods I talk about are prediction methods. Even the experimental ones I get to later today, they make mistakes. Okay? When you look at something, a result of that mistake, or a comparative model, the first thing you should find out is, where do you believe this is wrong? So according to some level of sequence identity, you can estimate, and I will show you numbers later, you can estimate the RMSD. So you expect an error. But you know that the RMSD will not be distributed all over the place. Which part of the structure that you modeled do you believe is most wrong? And why? Sometimes, in an ideal case, that could help you to maybe make a correction. Most often you cannot make a correction, but still, knowing this, doing this contemplation is very, very important to learn about your model. Okay? A similar statement here, and that is Thorsten Schwede, from who I got this. A model essentially is a tool that helps to interpret biochemical data. Now, what is a model? In some sense, I talked about comparative models. We are all clear, those are models. What is an experimental structure? I'll get back to that in a moment. And actually, model is my assumption for truth. It's just a degree of error, and that is shifting depending on whether I model it in the computer, comparative modeling alone, or I fit 3D data. Let me bring up the other talk. That was just the leftover of last week. Yeah, last week. It's not really the one. Yeah. Oops. So let's get into structure. In principle, I said, from Epstein and Anfinsen, we know that at least some proteins are such that we throw them into a solution and they adopt a unique three-dimensional structure that is important for the function of the protein. Now, that also implies that in principle we should be able to predict or the structure and to do it from first principles. And in fact, we can do that. As you all know, the structure prediction problem has been solved. There was a famous article in the Washington Post in the 60s that in fact explained how it had been solved. There was another article in the 70s, that just in the 70s it had been solved for the first time, this time in the New York Times. And there was another one in the 90s, it had been solved for the first time in the 90s, this time again in Washington Post. Those are all cover page articles. New York Times, Washington Post. As a scientist, you want to make a cover page of these. This is something, right? And now you see it has been solved 3 times. Something is fishy, right? And things get solved again. What's the problem? So obviously it's not right. And your background, you know it's not true. But it was claimed. It was claimed, again, these are not journalists who, it's not the Sun or something like that who just want to have some slander. These are serious journalists who are very careful, and serious scientists behind that, who are careful and check their models. How come they can have been so wrong? Don't go for they are idiots. They clearly were not. Yes?
So predicting the 3D structure of one protein from sequence alone is by far not being able to predict the 3D structure of a protein from sequence alone.
Well, if you do it by first principles.
My guess is they overlaid something.
Yes, now getting back to the 60s, we are not talking about a computer that can overtrain. In fact, this one here was also, I'm not revealing names here, but I can tell you that none of those was heavy on the use of computers. That, in some sense, this solution here was the most computer intensive, the middle one. So can you overfit?
Maybe the space of known proteins was so small at that time that it was possible to predict all of them, but it's just not the entirety of all proteins.
It is. That is a very very good point. But in this particular case, I don't believe it's true, but it could absolutely be true, and we get back to that point again. Sorry, I have so many references today, but I believe it's really more along the line of what Nico says, that we overestimate or we overfit because we were not carefully enough doing the cross-validation. How would you estimate structure prediction? How could we avoid that? Yeah?
You would probably always have to use the known structures and then try to predict.
Then you run into the same problem, right?
Right, exactly.
CASP? Isn't CASP just about this?
CASP is just about this. Have you all heard about CASP? The critical assessment of structure prediction. So essentially the idea is we have organizers begin to collect experimental structures. So what they do here is they talk to colleagues and ask them, are you going to work on a particular structure that you haven't finished yet, that you will in fact finish over the course of this summer? Okay, we line up 100 such cases, 100 structures that in the first CASP really came from as many groups, and in later CASP came from fewer groups. Then there is a prediction season, June to August. The prediction season stops at the moment that somebody outside the lab that does the experimental structure sees that work. So that could be the manuscript is submitted, okay? Could be other ways of publishing it to other people. Talk is given. That is the deadline. So everybody has to by then send in their predictions. Then there is a period here where assessors divine, I write. So essentially they look at the models and come up with a conclusion how good they are. And then there is a meeting to discuss the results. The person who initiated all of that in '94 is John Molt from Carp, Maryland. Initially very much helped by Tim Hubbard, who's at Sanger. By Christoph Fidelis, Richard Judson, that was the first, and Jan Peterson. The first group who in fact ran and organized CASP. Let me use this, I don't know, but I'm just going to deliver a personal grievance here. It's so complicated to find photos of you guys. So it would be very, very helpful sometimes if somebody sends me an email and my brain doesn't work and I cannot remember having seen that person. Then I Google and I never find.
We're not famous.
But there must be photos of you. Whatever. Just do more of an effort to put your stuff.
My name's Nikos Papadopoulos. There's literally 500,000 people with that name.
Now that's a good way of avoiding that anybody's ever going to find you. Just like Smith or something like that. Yes. So change your name. And how can you enter science? Just turn around 2 letters. It's so simple, I know. Well, you find a way to change the first vowel or something. So later, Andrei Krzysztofic came on board, and Anna Tramontano from Rome. At some point I was a co-organizer too. There are different categories. So one category is really tertiary structure, initially dropped after CASP 5. Secondary structure prediction of structure complexes was only done once. Later went into a similar competition called Capri. Residue contact prediction started in CASP 4. Disordered regions starting CASP 5. Function prediction starting in CASP 6. Model quality assessment. So essentially how good can you assess that the comparative model you built is right? Or that whatever you do is right? It's a different discipline. And model refinement, so that actually is the Swiss model versus modeler initially. Can you improve over the template?
Just a question, at what number are we right now?
10. And yeah, my slides are not good. So the first conclusion is there's no successful prediction of 3D structure from sequence, right? We have some accurate 3D predictions for cold proteins, but we can recognize the incorrect 3D structures increasingly successful. But can we extract principles about structure formation from structure? Well, to some extent we can. But in fact, is where we improved, and I'm going to get into threading on this. Clearly, the structure prediction servers that came out of CASP have impacted the field. One thing, so that's the first CASP, 1, 2, 3, 4 every 2 years, started in '94, that brings us to 10 today. The number of groups and the number of predictions shoots up over the first 10 years of CASP, so over the first 5 CASPs. And again, what we really can do is comparative modeling. And at this point that I did that slide, I still called it homology modeling. But there again is no general prediction of 3D structure. There are important improvements overall. The most important improvement is that we can push this one much further. So in '94, we found way fewer and made way more mistakes in finding and identifying the correct hit. This is way better today. That's the major improvement. Okay. Another thing was that in the first CASP, sort of everybody did their own method. Okay? Because, you know, how would you trust the one sitting next to you? They're idiots. You're the only clever person in the world. This is the way scientists operate. And somehow it's very, very careful, very difficult to avoid that. I believe, I would argue you have to find a way to avoid it. But everything that you're taught is against this. Everything you're taught has to do, and I'm contributing to this, clearly. I want to teach you to find mistakes, errors in what other people do. Not where they are right, really. That is, you know, left for your own capability. And somehow that sort of trains the dissection more than the unification of things. And I believe that's one of the problems. But then in the first CASP there were big time losers and big time winners. And that led to CASP 2. So immediately servers shot up, and in CASP 2 people began using what had been the winner of the first CASP. And that got us into prediction servers and meta servers. That is in fact one of the, and then the other state, in fact, to have people who use these machines and improve over the machines. What CASP assesses, and all the numbers I'm going to show, or flash, I'm going to flash some numbers, always are the prediction of a single person. A typical prediction season in CASP has 100 targets predicted. If you lose CASP, you don't get the grant next time. In the US, the situation is as dramatic as that. That means when, you know, in the US, a typical R01 grant is $250,000 for 5 years, or used to be in the olden days. So we're talking $1 million spread over 5 years. So just spend 3 months to get it or not get it. So you get some idea how much you're going to put into this. Okay? And nobody relies on the machine. So they do whatever they can do 24/7 to get the best prediction with whatever intuition they have out. Become a winner at CASP, get the next grant. That really has become this situation, and that is in fact one of the issues why machines or humans have learned to use machines and have learned to combine methods. That's great. So that clearly brought the field in many ways together. So the numbers I show are largely from CASP 9 in 2010. And in CASP 9, the main categories are template-based and template-free and model refinement. Template-based means essentially, like in comparative modeling, there is a structure that you can look up that is similar, only you have to find it. Template-free is you have an fragment assembly without the fragments, so to speak. So you create your own fragments. In some sense, that's essentially the idea. Or the idea is that once the structure is there, you don't find anything in the PDB that is similar enough so that you could have used it as a template. Model refinement, again, is simply you look at the template you found in the first category and you try to refine it. That's comparative modeling, threading, analogous fold based, and these are really sort of the de novo type of prediction methods. The quality measure is essentially how good you fit in terms of a 3D structure, and this fit is not an RMSD because there are too many too many problems with that. Template-based modeling, what this one shows is target difficulty. Typically that has to do with how much the target diverges from the template. Higher levels is residues that are matched at some proximity in 3D fit, so you want to be at 100. The different lines are for different CASPs, so again there are 9 CASPs shown here, and you see some tendency that for later CASPs this line is higher. Okay? And then there are overlaps. But overall, over time you see 2 events. The line shifts up, and you clearly sort of do way, in some sense moves up most here for the more complicated ones, because we can in fact find targets, identify targets better. Again, mostly the methods, so in CASP 1 I said there were losers, and the trouble between CASP 1 and CASP 10 is that in some sense method development is not really encouraged. What is encouraged is that you get the method right, not that you get an original method that brings something that nobody else brings. So that means that a lot is somehow circling around. Everybody tries what everybody else does, what has worked so far. That's one of the problems in the field. Again, meta servers, meta servers. This is just sorting here by a name of a protein, and that's the fraction of the predictions for which, in fact, that can improve over the template. And so the 0 line is no improvement. So these guys here, they improved in comparative modeling. These guys made it much worse. And somehow this is 2010, the CASP in 2010. And you see what in 2010, I said Swiss model was developed in 97. Manuel Peitsch said you don't do much, you don't do many mistakes. 13 years ago, he was scared about this imbalance here. And 13 years later, this imbalance is still dramatic. Despite everybody using the tools that work, despite everybody trying to copy as much as possible from meta servers, the vast majority of things still do worse with respect to just copying the coordinates. Again, it's no longer that trivial. Now finding it requires much more than just running BLAST. That's hidden in this slide here. But still, once you have found it, whatever trick you apply to find it, once you have found it, most of the time you make things worse once you try to improve them. Okay? Okay, now the template fit is crucial. You cannot get away from mistakes you make in the beginning. Let's get over the binding site prediction. I'm going to skip. I feel like I'm skipping some stuff today. Problems with CASP. Now let's begin with something like this. Assume in a particular CASP season, and that's not an unrealistic example, we have 100 proteins. Given that you have 100 targets, and given that some experimentalists may not deliver in time, you will not get structures for all of those in time. Some set, let's say 70% or something like that. Now I have a method A that uses 53 proteins and reaches a performance of X. I have a method B that uses 35 proteins and reaches a performance X plus Y. Is B better?
It depends.
On what?
Why not divide performance by proteins? And see how
oh, no, performance is per protein or an average. So this is not a number that is cumulative.
Okay.
That's a percentage of right. Or, I just didn't want to give any number, particular value. GDTS, as I showed before, so the residues that fit. Let's call it, just for the sake of the argument, X is the percentage of residues where the model, essentially by some criterion, built the correct model. It is within some proximity of the real structure. And again, this is a per protein average. Yeah?
Usually, isn't it when you use less proteins, you have a higher chance of overfitting anything?
That is certainly true, but we are not talking, mind you. So we are not in the Washington Post issue here. All of these predictions have been done on structures that were not available.
Okay, so it's
CASP.
All right.
It's true. So, I mean, I'm not. Let me be more explicit. I'm killing your logic here. I'm not killing what you say, because they're.
Maybe it's not so much overfitting as if you only test on a smaller set of proteins, you have less chance of getting one that your method doesn't work on.
So then that should clearly be better.
No, because they only.
You said a smaller chance.
And they have.
But you say if you have a smaller number of proteins, you have a lower chance of getting something that works. Isn't that what you said?
It doesn't work.
Oh, that doesn't work. Well, so
yeah,
is that a statement that is strictly true in a statistical sense? Yeah.
I don't know the answer of that.
But you have a different question, okay?
But I would say, yeah, it's just, I would say the 35, if you pick them clever, and you only take the 35 where you have a really good homologous protein, then you're gonna be better, of course.
So there, I believe you
cheating when you don't take all of
okay, so you make 2 points here. One, I believe, is very valid and very important. That is, if you know that your method is not as good for some, and you leave them out on purpose. And that may be sort of cheating. And I agree, for instance, let's look at comparative modeling. We take only templates where the sequence identity is very high. And we immediately would recognize this as cheating. On the other hand, if you really could look at a model and say, this model is better than that, and both of those were taken from 60% sequence identity, or 20%, or whatever that is, then that actually would be a power of the system. Right? And then, if you then could say, well, I'm only going to select the best ones by my own method, then that actually would be a virtue, would be something that should be rated as good, right? So back to the story that I made in the with the Marguerite pipe. So if you can in fact look at a model and say, here I'm wrong, here I'm not going to look, then that is a good feature, right? So this is unclear. What else? Well, one issue, of course, that I feel we should stumble immediately over is 100 a good number? Is 100 enough? I pressed the wrong button. The other one is Y enough? So is the difference enough? How would I answer that question? In this particular case, 35 and 53. Yeah?
I wouldn't like only having 100. I'd like having much more, but there's not really much we can do about it.
True.
Is there?
No.
So I guess we just have to work.
The question continues to be, is 100 enough to make statements like that? How is 100 enough and is Y enough? What decides on the answer to that question?
Yeah, you should try to have the distribution.
Exactly.
And then you can see if it's significant or not.
So if that would be A and B, then the difference is enough. If that is A and B, then the y difference may not be enough. So it really depends on the distribution and the value of y with respect to the distribution. So that's one very, very important point that you have to take into account. But let's get back to that issue. You already said that 35 is a smaller number than 53. But the point is, I never made the point that they are a subset, right? Is it okay to compare the different sets? Do you have to demand that at least the 35 is a subset of the 53? Yeah?
It depends. Maybe they are both aimed at different kind of proteins.
Yeah. So one does alpha helices and one does beta strand. Can I then say that they differ? Even if I had this difference here?
It's more this comparing apples and oranges a little bit, right? Because they're completely different.
And that is totally wrong, right? We clearly should not compare apples and oranges. Absolutely true. And then there is an issue about ranking, inappropriate ranking, and too few targets. But let's talk about something. Let's just assume, I'm going to show you a slide on the next, data on the next curve that is mildly put slightly confusing. And you don't really have to understand the data, but I want you to understand the message. Now the background is the following. I have 7 methods that I compare. And for these 7 methods, I have 1,000 proteins. So in this particular case, whatever the background, let's not talk about it. I have 1,000 proteins because 100 was not enough. And now I assume that in fact I sample from these 1,000 and create different pseudo-CASP experiments. And I'm asking, what gives the best ranking? And here's the first alternative. I take 29 proteins that are different. So every method is assessed on 29 proteins. They are sampled from the larger set. And the second case, I sample 11 proteins that are identical. What is going to be better?
Obviously it's the first one, because the variance is higher, so you have a better chance to distinguish different methods.
Anybody? I very much love your answer. In particular, the obviously part about it. Yeah.
Well, if the protein we're trying to predict is of the same as the same family as those 11 identical ones, then would be better in those.
I can't remember. So I said 1,000. I had 1,000 in the background, but I believe I did this experiment really on 100. So I sampled them from 100, and those 100 were sort of a random distribution of 3 months of CASP, or 3 months of structure determination, which is reflecting of what structure can do, or structure determination, experimental structure determination can do today, but it's not sampling all alpha, or it's not sampling all beta. In that sense, it's not biased. Okay. The other reason why I like your answer is because this was absolutely what everybody at CASP for many years believed is true. This was the predominant belief, and you see from the way I describe it that I'm going to show you the opposite. But this is what everybody in the field had assumed for many years, your answer. Because 11 and 29 is a difference of 3 times. If you say 100 is small, but 11, come on, is 1/10 of it, right? So 11 identical, so they're 7 different methods, 29 different. And each dot here is 1 CASP. And then, I do just 30 different CASP or something like that. And I ask, in fact, I have the results for 1,000 proteins for these 7 methods, so I know, in fact, what is the true ranking. And I'm asking how much does the ranking that I get in one of these 30 CASPs differ from the true? Hi, come in.
Sorry.
Don't worry. I'm sorry that the door is closed. So I can simply compare in this 29 different, in this scenario 11 identical. How close is my ranking here to reality? How close is it here? And what I find is that this is much, much worse. In fact, in this scenario, every method becomes the winner of a CASP, even methods that are clearly the losers. But just in a sample set of 100, and in some sense that is what the two of you implicitly said, the probability that if your sample set is large enough, that you luckily sample the right one is just high enough for one particular CASP. And CASP is one frozen picture. Okay? Now, Again, if you look at the average of all of these, you again evolve a picture. But what we get at CASP is just one shot, every single CASP. And then in 2 years we have another CASP, but that is in fact a new method. Because we all try to do better next time, right? That's one problem. How can I sort of find a path that joins these so that tries to have as many proteins as possible, yet always compare on identical sets? How can I do that? Optimize somehow the realization of both objectives. Any idea? So you essentially look at pair comparisons. So you compare method A with B, and in that comparison you use whatever they have in common. Then you compare A with C, whatever they have in common. That gives you the opportunity to in fact have here a higher number than here. So use whatever you have. And still the information here is that significantly better by the distribution background according to the set we have. And the same thing here. So essentially what you enter into the table is significant improvements. And then you sort of optimize the number of things you compare. So you get as close as possible to the 29 as often as possible, and yet have similar sets. And then even in that one there are some issues. But these are issues. Some other issues are in fact that CASP is very much biased by the way people look at it. Danny Fisher set up CAFASP, that was the automated CASP. CAFAS went into two different things, LiveBench, that was set up by Danny Fisher, Arne Elofsson, Leszek Rychlewski, and that in fact LiveBench is an automated testing of servers. And another person here is Laszlo, who was in the group, you may have seen him. There's Eva, that's a similar about words. How do you pronounce that?
I pronounce it Eva.
That's good. Good. Eva, right? Obvious.
Totally.
Huh? But you all take part in St. Patrick's Day. You should be able to recognize Eva here in this Irish name. So Eva is in fact the same thing. So the idea is that you pick up from PDB things that are not quite released yet or not embedded into servers. You run with the sequence servers, then you sort of test and you publish every week results, and you keep running that for many years. And again, it is sort of in some sense an automated CASP. Volker Eirik, André Schalli, Marty Rohn were involved in that, and I'm going to show you some of the, another person was Ingrid Koh and Guy Agdaf. And I'm going to show you some of the results from Eva. Unfortunately, Eva is no more. Here's one. It died the timely death. Sequence identity versus RMSD for different modeling servers. Swiss model, modeler is not in here, and a few others, 3D Jigsaw is for instance in here. So the first thing you see is that comparative modeling is sort of fairly accurate. Here it's something that corresponds to 2.5 angstrom RMSD until almost 40% sequence identity, right? And this now is a test on, I believe, this is based on 10,000 models, or 10,000 cases. This is a huge data set that Eva collected over many years. Let's go on. Now, the unfortunate reality, or the reality of a typical PSI-BLAST run, if you plot the identity distribution, you cut off at a PSI-BLAST 10-3, and you take whatever you find at 10-3, and you ask, what's the mutual sequence identity, and what's the distribution of that? Then you get the red one. Telling you, yes, sort of models are good here, but most of the things you find are more in this realm. Okay, that's the first sort of slightly negative way of looking at it. The second slightly negative way of looking at it is percentage sequence identity again against coverage. And what this coverage means is now, here is the point where I said under 40% sequence identity here, you have roughly 2.5 angstrom, so you produce very good models. But actually, these models cover only about 60% of the protein. So the comparative models that you build, I skipped that statement, but for instance, Swiss model currently builds for 1/3 of UniProt. A model automatically is there, is in the database already. But that means that for some fragment in this protein, for 70% of that fragment, you find something that is acceptable. That's good and bad, right? And again, most things you find are down here in the realm of where you in fact have hits, but they're actually relatively short fragments and error prone, okay? Comparative models are great and they're the best thing we can have, but still there are issues. Now, the twilight into the midnight zone. So again, I said up here, the speed of modeling is the problem. Down here is the detection of the homology. And again, so that is this region down here. Can we get into the midnight zone? Again, the statement here is most structures of proteins that are similar are completely unrelated in terms of sequence. It's the midnight zone story. And going from the safe zone or daylight zone into the midlight zone, we go through the twilight zone. In the twilight zone, we go from 100% correct, this is for pairwise alignment, to something like 10% correct. So there's a phase transition. Again, we have a finding problem. But what else can we do in order to get into that? And that is now, at least two people in the room have brought up this issue. It goes back to Bowie. Jim Bowie is one of the people. Fit sequence propensities into a 3D structure, commonly known as threading. That's the term coined by Janet Thornton. Fold recognition. Essentially, the point is, now once you have a 3D structure, you can reassess. You can reassess because the 3D structure implies some proximity, some relations between residues, and they may not fit. That is the entire idea behind threading. You somehow define an energy function and you move things around to optimize this energy function. And then again, there are different ways of implementing that. This is the one that coined the term, David Jones, Janet Thornton, that coined, Willie Taylor, that coined the terminology threading. You thread the sequence through the 3D structure and you ask how well do I fit in a particular point. I also showed you, so that's the threading idea. Now you could in fact alternatively, instead of aligning into potentials, align secondary structure. Predicted secondary structure or preserved secondary structure is a similar idea. And in fact, all of this is now combined, and in fact the large breakthrough really is that we can push the twilight zone and go much, much deeper. That improves comparative modeling. We find better, we do better. That's the big breakthrough. What will be the next step in terms of intruding into the midnight zone? How can we go deeper? We can use structural information, that is threading, and all those kinds. We can use fragment approaches, we can use modeling approaches. That's all part of it already. What could we do beyond that? Go out and jump. Well, profile, we talked about it already. Nonlinear scoring, avoid homologous overextension. Those are sort of small fixes. Ultimately, the answer is, short of being able to really model a structure, your answer is as good as any. Or the one that you just presented to me in speaking, shouting out, meaning there is no big answer. So the major point is we are back to sequence alignment. So the 90s and 2000 was very much about using the potential of improving the alignment, because I know the three-dimensional structure from the comparative model. And now we are really back, and we fixed, that has been fixed, that has been done. There's a great improvement. This is one of the main steps. Another step is to in fact use low resolution experimental information to improve the model. Overall, the best way to predict structure is comparative modeling. As good as the template, depends on the quality and the similarity to the template. Sometimes we can do a little bit better, but not much, mostly driven by the accuracy of the alignment. In the loop modeling, we're still not there. Sometimes we can do it. And in the sidechain modeling, it's in fact not quite clear how well we do. There are some successes, and we numerically can assess sidechain modeling, and it appears to be very accurate. But the problem is that we make too many mistakes in the main chain, some people argue. So when the model is that inaccurate in the main chain, there's no point in assessing sidechain accuracy. That's the debate here, and that debate is in principle not over. Where do you believe that you do? I'm going to wrap up today. I have an example, but I don't want to go through this example. Somehow I feel it's not the day for that. The example of Eve. And the example of Eve is a story that, let me just present the part of the story that comes from Marco Punta and Claudia Bertonatti. She's no longer at Sapienza, and is she I can't remember. Maybe, yes, she's in Paris, I believe now. And it's a story about sequence, structure, and function. Comes from structural genomics, and that in fact is something that I'm going to talk about next Tuesday. Is an example where we have a lot of structures for something that is very, so rather than in the story of comparative modeling find one template, in this particular case we have 6 structures. And so 6 structures new. We had 4 before, and now we have 6 more. And we have a lot of sequences. But we don't know what this thing does functionally. And the whole story is about how we can use structure to learn some ideas about the function. But I'm not going to go through this. My question to you, so we do, comparative modeling, it reaches some level of accuracy. Now, is there any, so I talked about the pipe. I believe at some point I have the pipe in here. I like Magritte. So is there any point in having a model that is wrong?
Depends how wrong.
So what kind of thing could you imagine you learn from it?
Yeah, if you know, you can have a perfect model, as I said, because then it couldn't be a model anymore. But if you know where your weaknesses are, then you can use it to your advantage.
Yeah, that's true. Now,
so you would want a model that is wrong at the right places.
So what are these right places?
If you maybe have, like, you say you have 2 domains and something that connects them, and the domains you model more or less correctly, but the connection not, then you can already know something about the function. Because you know what the domains do, and then maybe something like this.
So a connecting loop. Now, let's get back to exactly that image you have in your head. I do a comparative model on a protein that has 2 domains. I simultaneously model these 2 domains, and in some cases I make a mistake because the target and the template differ very much in the connection of the 2 domains. How realistic is that scenario? And I understand by you phrasing that scenario, and you, by the way I make the statement now, you understand that it's not very realistic, and that is something that I did not make clear. So comparative modeling, for all it works, is in fact to model into domains. So in fact, having 2 entire domains and model those, oof, yes, there are examples. And there are particular structures where in fact that is more likely to work, but this is on average still the exception. Now today I told you most proteins in PDB are single domain proteins, while most proteins in SwissProt or in UniProt are multiple domain proteins. So maybe that statement is going to change when PDB moves into becoming more of a multi-domain database. Could be, I actually don't know. But today modeling really is on single domains. So while you're right at what you're saying, I'd like to rephrase the question. So let's assume I have a model for a single domain protein. What are the cases, or what are the regions that you want, or that you could accept to be wrong, and what are the ones, yeah?
I mean, if we get the catalytic center right.
Good. Now, do you believe, if I sort of looked at the catalytic site and I compared the model accuracy for a set of enzymes for the catalytic site and for everything outside the catalytic site, how do you believe they would differ? Would the catalytic site be more often right or more often wrong?
I would say more often right, because...
Why would you say that?
It's better conserved, maybe, and then you find a good... Because it's not working when there is a mutation in the site. Or maybe...
So it's a functional selection. That is in fact one strong argument why this expectation is a good one. I believe we are still lacking a very, very clear answer to that, but the data that has been presented so far indicates that your suspicion is right. That in fact active sites, enzymatic catalytic sites, or various functionally important sites are more conserved than the average. Okay? That's statement number one. Let's have the next statement. So you're totally right that what I want to be more right in the model with is the functional site. And the functional site, luckily, on average, is more likely going to be correct. Let's get, yeah?
Is it more likely to be correct? Or is it more, I mean, I can understand that functional site being conserved among many proteins, but why would it be more correct in my model?
Again, so the point is, don't forget, the model is, my statement was the model is as good as the fit between target and template. So if the most conserved part between target and template is the functional site, then that, and again, more likely is simply, I'm making a statement that has nothing to do with statistics, it's just number. More often, I should say. It's more often better predicted because it's more often conserved than average. That's good news. So now let's turn the story around. So which part of a structure is least likely, again, sorry, when you look at a protein, which thing would you look, how would you describe, I'm telling you there's some aspect of a protein or some region that has a lower average than the other. What could that be?
Surface loops.
Exactly, in some sense what you said before, Verena. Surface loops, yes, so loop regions. But now, if we put these two things together, we have a problem, right? Because think about an antibody. The entire function of the antibody is the long loop that you have. And the way you in fact fix that long loop to adapt to different substrates. That in fact is what your immune system learns. That's what you do. And so there you have an example where something is less conserved because of functional reasons, where you're more likely, in fact, again, not more likely, I'm sorry, where you will make more often mistakes, and it's functionally important. So that, in fact, is one of the reasons why the statement that you made before, Verena, or why this is the active site, the functionally important site, better predicted or not, still has an issue, because there is not one solution. But at least this is a good model. There's a wonderful story that Anna Tramontano has been traveling with for many years. She produced, when she was at a company, Merck, in the south of Rome, she produced a model, and that model led to the development of an important drug for the company and for her, and the model was totally wrong. And it led to this drug not because it got the functional site right. It led to the drug because it got an idea how to change the functional site. And that idea was right. They did not know. She did not know when she started that the model would be that wrong. She believed the model. But the aspect of the model that she pushed was the one that led to the development, and that single bit was right. So sometimes you can be lucky, is the way she tells the story. I believe it's more than luck, because she has repeated things like that. And at some point luck doesn't explain this sort of thing. Now, I believe there was something else I wanted to make, but I forgot the point. If there are no further questions, then we are through today. We will talk next week about, there's no lecture on Thursday. We will talk next week about structure determination and 3D to 1D secondary structure assignment, and then move on to secondary structure prediction. Thank you. Any questions?